Programmed cell death protein 1, also known as PD-1, is a 288 amino acid cell surface protein molecule that in humans is encoded by the PDCD1 gene. PDCD1 has also been designated as CD279. This gene encodes a cell surface membrane protein of the immunoglobulins superfamily. This protein is expressed on T cells and pro-B cells and plays a role in their fate/differentiation. Structure: Programmed death 1 is a type I membrane protein of 268 amino acids. PD-1 is a member of the extended CD28/CTLA4 family of T-cell regulators. The protein structure includes an extracellular IgV domain followed by a transmembrane region and an intracellular tail. The intracellular tail contains two phosphorylation sites located in an immunoreceptor tyrosine-based inhibitory motif and an immunoreceptor tyrosine-based switch motif, which suggests that PD-1 negatively regulates TCR signals. This is consistent with binding of SHP1 and SHP2 phosphatases to the cytoplasmic tail of PD-1 upon ligand binding. PD-1 is expressed on the surface of activated T cells, B cells, and macrophages, suggesting that compared to CTLA4, PD-1 more broadly negatively regulates immune responses. Ligands: PD-1 has two ligands, PDL1 and PDL2, which are members of the B7 family. PDL1 protein is upregulated on macrophages and dendritic cells in response to LPS and GM-CSF treatment, and on T cells and B cells upon TCR and B cell receptor signaling, whereas in resting mice, PDL1 mRNA can be detected in the heart, lung, thymus, spleen, and kidney. PDL1 is expressed on almost all marine tumor cell lines, including PA1 myeloma, P815 mastocytoma, and B16 melanoma upon treatment with IFN-I-cubed. PDL2 expression is more restricted and is expressed mainly by DCs and a few tumor lines. Function: Several lines of evidence suggest that PD-1 and its ligands negatively regulate immune responses. PD-1 knockout mice have been shown to develop lupus-like glomerulonephritis and dilated cardiomyopathy on the C57BL/6 and BALBC backgrounds, respectively. In vitro, treatment of anti-CD3-stimulated T cells with PD-L1 results in reduced T cell proliferation and IFN-I-cubed secretion. Reduced T cell proliferation correlated with attenuated IL-2 secretion, which can be rescued by addition of cross-linking anti-CD28 antibodies or exogenous IL-2. Together, these data suggest that PD-1 negatively regulates T cell responses. Experiments using PD-L1 transfected DCs and PD-1 expressing transgenic CD4+ and CD8+ T cells suggest that CD8+ T cells are more susceptible to inhibition by PD-L1, although this could be dependent on the strength of TCR signaling. Consistent with a role in negatively regulating CD8+ T cell responses, using an LCMV model of chronic infection, Rafiamida Euro unregistered trademark S group showed that the PD-1-PDL1 interaction inhibits activation, expansion and acquisition of effector functions of virus-specific CD8+ T cells, which can be reversed by blocking the PD-1-PDL1 interaction. As CTLA4 negatively regulates anti-tumor immune responses, the closely related molecule PD-1 has been independently explored as a target for immunotherapy. The 2-CTCR recognizes the peptide cyareagle in the context of H2 kilobytes 2-CCD8 T cells incubated with IFN-I-cube treated B16 targets expressing cyareagle peptide poorly lyse their targets and secrete low levels of IL-2. However, PD-1 knockout 2-CT cells have heightened cytolytic capacity and IL-2 secretion, suggesting that PD-1 negatively regulates anti-tumor CD8 T cell responses. Similarly, P815 mastocytoma, which does not express PD-L1 unless treated with IFN-I-cubed, can be transduced to express PD-L1, resulting in inhibition of in vitro CD8-mediated cytotoxicity and enhanced in vivo tumor growth. In vitro cytotoxicity and in vivo inhibition of growth can be restored by anti-PD-L1 antibodies or by genetic ablation of PD-1 together. These data suggest that expression of PD-L1 on tumor cells inhibits anti-tumor activity through engagement of PD-1 on effector T cells. Expression of PD-L1 on tumors is correlated with reduced survival in esophageal, pancreatic and other types of cancers, highlighting this pathway as a target for immunotherapy. Said et al. Showed that triggering PD-1, expressed on monocytes and upregulated upon monocytes activation, by its ligand PD-L1 induces IL-10 production which inhibits CD4 T cell function. Clinical significance: in mice, expression of this gene is induced in the thymus when anti-CD3 antibodies are injected and large numbers of thymocytes undergo apoptosis. Mice deficient for this gene bred on a BALBC background developed dilated cardiomyopathy and died from congestive heart failure. These studies suggest that this gene product may also be important in T-cell function and contribute to the prevention of autoimmune diseases. Cancer: monoclonal antibodies targeting PD-1 that boost the immune system are being developed for the treatment of cancer. Many tumor cells express PD-L1, an immunosuppressive PD-1 ligand. Inhibition of the interaction between PD-1 and PD-L1 can enhance T-cell responses in vitro and mediate preclinical anti-tumor activity. One such anti-PD-1 antibody drug, nivolumab, produced complete or partial responses in non-small cell lung cancer, melanoma, and renal cell cancer, in a clinical trial with a total of 296 patients. Colon and pancreatic cancer did not have a response. HIV, drugs targeting PD-1 may augment immune responses and/or facilitate HIV eradication. See also, pembrolizumab approved by the FDA in September 2014 to treat metastatic melanoma. References. Further reading. External links: PDCD1 protein, human at the U.S. National Library of Medicine. Medical subject headings. This article incorporates text from the United States National Library of Medicine, which is in the public domain.